We are now going to proceed to talk about important monosaccharides. The most important monosaccharides include glucose, galactose, and fructose. We are going to talk about each of them in detail. Starting with glucose. Glucose is a monosaccharide that is found in fruits, corn syrup, and honey. It is an aldohexose with chemical formula C6H12O6. We can see in its structure on the right side of the slide that it contains an aldehyde and 1, 2, 3, 4, 5, 6 carbons in length. That's why we know it as an aldohexose. Whenever blood sugar is being measured, glucose is the monosaccharide that is detected. Another word for glucose is dextrose. And glucose can be placed, as you can see in figure 11.23a, as a solution that then can be delivered to the human body via injection in veins. Glucose is an important building block because there's other disaccharides that contain glucose in its structure. Some examples include sucrose, lactose, and maltose. Additionally, glucose is the building block of polysaccharides like cellulose and glycogen. Next, let's talk about galactose, which we can see its structure on the right side of the slide. When it comes to galactose, galactose is also an aldohexose of chemical formula C6H12O6, and we can see its aldehyde and additionally the number of carbons, which are 1, 2, 3, 4, 5, and 6. Galactose is actually not found free in nature, meaning that you don't see galactose as a monosaccharide available in nature. It is actually produced from glucose. And specifically in human, what we see is that this monosaccharide galactose is produced from glucose in order for the mammary glands to secrete the disaccharide lactose because lactose is produced additionally because of the linkage of a galactose molecule and a glucose molecule. So the body has to produce it. Lastly, we are going to look into fructose. Fructose, which we can see in the last structure on the right side of the slide, it is actually found in fruit. Fructose is obtained from sucrose via hydrolysis. Fructose, when we look at its structure, it is actually a ketohexose. And when it comes to its chemical formula, it also shares the C6H12O6 chemical formula. We can observe that the ketone in fructose is up here. That's why it's known as a keto or a ketose sugar. And it has 1, 2, 3, 4, 5, 6 carbons. So it is still a hexose. Interestingly, out of the monosaccharides, fructose, it is actually the sweetest carbohydrate. It is known that it is almost twice as sweet as sucrose, which is a disaccharide. Continuing with the topic of important monosaccharides, now I'm going to introduce to you a specific monosaccharide that is found in another biomolecule. This monosaccharide is called ribose. Ribose specifically is a pentose sugar, meaning that it has 5 carbons in length. It happens to be also an aldose sugar, even though it's not illustrated right here on the slides. In this image, we can actually see that the biomolecule known as a nucleic acid, which we are going to learn about it in detail in chapter 13, has as a basic unit a sugar, specifically ribose. Now, there are two types of nucleic acids. The two types of nucleic acids we can see in the bottom that are going to be RNA, which is ribonucleic acid, and we also have DNA, which is deoxyribose nucleic acid. So, the name of these biomolecules is actually defined by the sugar that is present in its basic unit. And while RNA has ribose, and we can see its structure right here, you can even see the 5 carbons that are present in the ribose. As I just mentioned, this in its open form is an aldopentose. We can see that in, for example, in the other nucleic acid, which is known as deoxyribose, the word deoxy refers to the position 2 lacking an OH group. So in the ribose structure, once in position 2, what you have is 2 hydrogens that is known as deoxyribose. As I mentioned, these type of biomolecules we're going to learn in detail in chapter 13. Let's now continue looking at the properties of sugars. In addition, we will be talking about some chemistry that pertains sugars. First, let's understand that all carbohydrates are solid at room temperature because of the many OH group present. Specifically, we know that monosaccharides form hydrogen bonds with water and they are extremely soluble. In the body, this solubility is what allows carbohydrates to be transported rapidly in the circulatory system. We can see right here in the bottom left figure that we have on this slide how this actually work once we have ingested carbohydrates. I'm going to zoom in so we look at this figure a little bit more closely. So when we consume food products that have carbohydrates, during the process of digestion, these carbohydrates are going to be broken down into simple sugars. Next, what we see is that these simple sugars are going to be absorbed via the bloodstream. Specifically, to give you the example of the most important one, here we have glucose being represented by these little cubes. And you can see the red blood cells, and also you see right there that they're traveling through the circulatory system. Now, upon absorption, what happens is that we see the simple sugars, in this case, glucose, being absorbed and entering cells. And this process is actually possible because of a hormone. We're going to learn about hormones in the next chapter. Specifically, the hormone called insulin is what allows glucose to get into the cells in the body. Once these glucose molecules have entered the cell, specifically, as you can see, we're using glucose as an example. What happens is that then the body can utilize it as an energy resource. In the body, we have to generate chemical energy in the form of the molecule ATP, which we're going to learn about it later. Let me write it here more clearly, ATP. And then if you have glucose that is not used as fuel because it is consumed or in the body it is present in excess, then is stored for later use as fat. As I mentioned before, fructose is going to be the sweetest monosaccharide or the sweetest carbohydrate in general. We can see that in table 11.2, we have the relative sweetness of sugars. In this table, what we observe is that table sugar, in other words, sucrose, has been standardized as 1.00, and then from there, you see that numbers have been assigned to the other carbohydrates. Notice that in here we can observe that lactose, which is a disaccharide, is less sweet than sucrose, which is table sugar. And we can observe that fructose, as we've mentioned before, is actually sweeter than even table sugar. Next, let's look at some chemistry, starting with the formation of sugar alcohols. Sugar alcohols are produced in the food industry as a resource of sweetness that do not provide calories to the body, meaning that sugar alcohols are not utilized in the body in order to produce ATP. Sugar alcohols originate from the reduction of the carbonyl group in monosaccharides. When we are naming sugar alcohols, all we're doing is replacing the OSE ending in your monosaccharide name by the ending ITOL. So the names of sugar alcohols end in ITOL. Because these molecules are sweet, they are used in many sugar-free products, including diet drinks, sugarless gum, and even candy. To give you an example of a candy that is popular, and it has actually very interesting effects on the health of humans, here I'm introducing the example of xylitol. Xylitol specifically comes from the monosaccharide xylose. Xylose undergoes a reduction reaction in which when you react xylose with H2, and this specifically is the catalyst, what happens is that specifically the aldehyde in xylose turns into a primary alcohol. And that's how we obtain the sugar alcohols. Now, xylitol is known specifically as a sugar alcohol that cannot be utilized by the bacteria that live in our mouth as an energy source. So it actually prevents bacterial growth and reproduction for the bacteria that causes cavities. So it has been suggested that xylitol, chewing it specifically, can help the prevention of cavities. Here you see specifically a company created by this young lady, and she actually made this company basing all her candies with the use of xylitol as its sugar source. As you can see, since 1963, the use of xylitol has been utilized for the prevention of cavities and is FDA approved. It is actually as sweet as sucrose, but the only thing is that in doses over 40 grams, xylitol may have laxative effects. Sugar alcohols are carbohydrate derives. And since it was created in the laboratory, it is also part of the artificial sweeteners. Understand that an artificial sweetener is going to be any molecule that has been created in the laboratory that gives you the sweetness of carbohydrates, but it doesn't have the caloric output, meaning that it doesn't produce and specifically ATP molecules when consumed. There are a number of artificial sweeteners that are currently in the market, including sugar alcohols. And here I'm introducing some of the common ones that we have commercially available, starting with sucralose. Its marketing name is going to be Splenda. When it comes to sucralose, sucralose specifically, similar to sugar alcohols, is going to be carbohydrate derived. Sucralose actually was synthesized from sucrose. The only difference between sucrose and Splenda is that specifically Splenda has one, two, three chlorines that were placed in the molecule instead of OHs. When we look at sucralose sweetness levels compared to sucrose, we can observe that while sucrose is 100 in value, sucralose is 60,000 in value. So as you can see, sucralose is very, very sweet compared to table sugar. And here in the graph, just to bring it back, you can see that xylitol and table sugar are the same value. Moving right on. The next one that I want to introduce to you is going to be aspartame, which its marketing name is NutraSweet. We can observe that aspartame is not carbohydrate derived because aspartame actually comes from linking together two amino acids, which is when we look more closely at the structure, the two amino acids present in aspartame are aspartic acid and phenylalanine. And at the end of that phenylalanine, a methyl group was added to form an ester. Now, when it comes to the sweetness levels, specifically for aspartame compared to sucrose, we can observe that since sucrose is at 100, aspartame is at 18,000. So it is another artificial sweetener that it is much sweeter than table sugar. Lastly, we have saccharin. Saccharin, which we can see its structure at the bottom of the slide. Saccharin, specifically, we can see that is also a non-carbohydrate derived sweetener because it doesn't come from sugar. When it looks at its structure, we can see a benzene ring, we can see an amide bond, and we can see a sulfonate bond. That last function group we did not study as part of our curriculum. Nevertheless, when we look specifically at the sweetness levels of saccharin compared to table sugar, in other words, sucrose, we can see that saccharin is at a level of 45,000. So these three artificial sweeteners that I presented, meaning sucralose, aspartame, and saccharin, have really high sweetness levels compared to other food products, and they are commercially available. Also, understand that some of these artificial sweeteners are actually banned in certain countries. Currently, in the US, these are not banned. Continuing on, looking at the chemistry for carbohydrates, next we're going to discuss the process of hydrolysis. Hydrolysis is defined as the chemical breakdown of a compound due to the reaction of water. In hydrolysis, our reagents include water in the presence of acid or water in the process of an enzyme. In the next chapter, we will be learning about enzymes, which is a type of biomolecule derived from proteins. The main point that I'm trying to illustrate here is that when we compared monosaccharides, disaccharides, and polysaccharides, The only type of carbohydrate that does not undergo hydrolysis is going to be the monosaccharides. If you have a disaccharide, it doesn't matter if it's in the presence of water with one of the two catalysts that we have, meaning acid or enzyme. In a disaccharide, upon hydrolysis, what's going to happen is that you will be producing the two monosaccharide units that are forming that disaccharide. In the case of a polysaccharide, because there's many units of a monosaccharide, you need many units of water. Similarly, we need a catalyst. And upon reaction of this polysaccharide and water, what we're going to see as a result is the hydrolysis and the production of many monosaccharide molecules, which were part previously of the polysaccharide. So understand that out of the carbohydrates, only disaccharides and polysaccharides can undergo hydrolysis.